Hello, and a warm welcome to today's Select Science webinar entitled Kinetic Characterization of Covalent Inhibitors on the PHERAstar. My name is Emily Adam, and I will be moderating today's presentation. This webinar covers biochemical strategies for characterizing covalent compounds in vitro, exemplified by the recent discovery of a new covalent inhibitor targeting the hitherto undruggable cancer target USP7. Today's primary speaker is Agnes Martin, a principal scientist in the Screening and Compound Profiling Group at Cancer Research UK's Therapeutic Discovery Laboratories, specialising in biochemical and biophysical assay development for small molecule hit identification and validation. CRUK Therapeutic Discovery Laboratories is the in-house CRUK Drug Discovery Unit. Just before I hand you over to our speaker, I would like to mention a couple of things. Please feel free to ask questions for the Q&A session at any time during the webinar by clicking on the Ask the Question button at the bottom of your screen. To view the presentation in full screen, click on the tab at the bottom right of the media player window. Finally, an on-demand version of this webinar will be available in a few days' time. So, without further delay, I'm delighted to hand you over to Catherine Wark, who is Applications Manager with BMG LABTECH, who will begin the presentation with a brief introduction of the PHERAstar microplate reader series as used by Agnes in her research, and then Agnes will present the scientific content on our session. Go ahead, Catherine.
Hi, my name is Catherine Wark and I'm an application specialist with BMG LABTECH UK. As Agnes today, we'll be presenting data that has used the PHERAstar high-throughput screening instrument from us for many of her screens. I will take a few minutes at the start of the webinar just to introduce this instrument as utilised by Agnes and the team. So, to start with, Agnes's team have utilised the PHERAstar FS, which is the foundation of our latest PHERAstar innovation, the PHERAstar FSX. The PHERAstar, launched in 2009, is a well-established screening product for high throughput and sensitivity, so very well established and the first choice for those working in HTS. This system allows users to read all non-isotopic read mode technologies in all plate formats up to 3, 4, 5, 6 well plates, and allows simultaneous dual emission where there are dual emission readouts such as Brett, fluorescence polarization, Brett, and absorbance assays with the use of our spectrometer technology for instant analysis of both spectra and multiple readouts. Optically speaking, the PHERAstar platforms use an optic module concept. We have matched filters in barcoded optical blocks for ease of use. Top and bottom reading for users doesn't require any sort of manual intervention. It's all controlled via software. And for the PHERAstar, one of the outstanding features has always been our HRF performance. We achieve this great performance with our matched photomultiplier tubes that are redshifted and photon counting specifically for this application. To move on from this and talk more on HRF performance, we also offer 2 excitation sources for HRF-based assays, or TR-FRET, a xenon flash lamp and a TRF laser. With the PHERAstar FS, this is a 40 Hz laser, and with our FSX is a 60 Hz laser. As you can see from the data on the right-hand side of the screen, whether using europium or terbium as a donor, as shown with the HRF reader control kit data, both read technologies give fantastic Z-prime data. You will, however, notice that on the right-hand side where we look at the terbium results, where the laser is utilised, because this laser excites specifically on 337 nanometers, we get an added enhancement to the signal to blank, whilst maintaining, again, good Z primes. So for this type of assay, the terbium type assays, the laser might be of particular benefit. The biggest benefit, however, of using the laser is the fact that we get much greater energy input with one laser flash compared to the xenon flash lamp, and this means for users that are wanting to increase throughput, reduce read times for say 384 and 1536, the introduction of this technology can make assays significantly faster. So to talk back again to more standard features of our instrumentation, we can perform Z height adjustment for both top and bottom reading, bottom reading being important for cell-based assays. We have 3 integrated barcode readers inside for readings from south, west and east, and of course a stacker for continuous loading of higher throughput plate formats. To introduce some other standard features of the PHERAstar FS and FSX, we have incubation and shaking. This is important when maybe measuring enzymatics or cell-based assays. And for those looking at cell monolayers, our high-resolution cell layer scanning can also be used, as shown in the bottom right corner, to be able to look at changes in signal across the well. For those working with HTRF or alpha technology, we also have a unique decay curve monitoring tool which is ideal for troubleshooting assays and optimizing the best signal to blank for this types of assay situation. Now, in addition to these standard features, PHERAstar FS and FSX can have 2 injectors built into the side of the unit, so whilst maintaining the same footprint. These injectors allow users to vary the volumes across the plate from well to well, changeable in as little as 0.5 microliter increments. Users can control also the injection speed when working with different liquid classes or cells, and for very rapid reads, we can read up to 100 times per second utilizing this system. So that covers in general the PHERAstar FS platform as Agnes would have used it, just to introduce some of the newer features that the FSX introduces. Again, while still within the same footprint of the FS, so for existing FS users, this is compatible with the platform they already have, if you look to increase on that. The FSX offers enhanced sensitivity in all of our read modes, with faster read times in particular for TR fret with the faster laser, the 60 Hz laser. We have a broader dynamic range of one order in addition for luminescence assays and some crosstalk reduction technologies, along with improved alpha performance based on speed and performance, along with the introduction of simultaneous dual emission for alpha technology to accomplish things like alphaplex readouts. So to summarise then, really both the PHERAstar FS and FSX are both a fantastic platform on which screeners can get the most of their assays. The injectors and the read and inject simultaneously, those sort of unique features of the PHERAstar enable even some of the most rapid kinetics to be measured. And with the FSX, for those thinking towards the future, you could look to see the same performance you see from your FS with additional performance enhancements in both speed and sensitivity with additional alpha function, for example. So at this stage, it pays for me to really pass over to Agnes to talk more about how this technology is used in her screening projects and in the kinetic analysis of the ubiquitinases. Thank you very much.
Thanks, Catherine. So the areas I'd like to discuss today are the resurgence of covalent compounds in drug discovery, the mechanism of covalent inhibition, assay development considerations for covalent compounds, various biochemical assays that can be used to triage covalent compounds, various biophysical assays that can be used to profile covalent compounds, and I'd like to go through the discovery of a covalent inhibitor that we've done for the cancer drug target USP7. Covalent drugs have a cysteine targeting element which links them covalently to their target. Many examples of successful and effective covalent drugs exist. However, historically, these tended to be serendipitous discoveries. The drug discovery industry actively avoided covalent strategies due to safety concerns. recently covalent strategies are increasingly considered, and it's now recognized that carefully tuning the reactivity of the warhead and relying on specific complementarity to the target can yield a number of advantages. These include prolonged duration of action, which results in less frequent drug dosing, high biochemical efficiency, which results in lower doses, non-equilibrium binding, which allows us to overcome competing endogenous substrates, and it also gives us the possibility to address targets with shallow binding sites. Against that, a hyperreactive warhead might lead to drug-induced toxicity. Covalent strategies are not suitable for mechanisms requiring a short residence time, and little advantage has to be gained for biological targets that are turned over rapidly. There are a number of considerations to take into account when developing assays specifically for covalent compounds. Because covalent compounds have a warhead that reacts with a cysteine thiol group, this has an impact relative to common reducing agents that we use in vitro assay that also rely on a thiol reactive group, and these include glutathione, beta-mercaptoethanol, and DTT. These have the potential to compete with the target cysteine, especially when you consider the relative concentration of the different reagents and the fact that the reducing agents are in vast excess. We found that this can lead to false negatives. An example here is a set of compounds that were tested in two assays with identical buffers except for the reducing agent. One assay had DTT and one assay had TSEP. Reversible compounds gave a good correlation between the two assays. However, covalent compounds were not detected in the presence of DTT. This is likely due to the fact that TSEP has a phosphate as a reactive group and not a thiol, and so it's worth bearing in mind that using thiol-based reducing agent can lead to false negatives. Another consideration with reducing agents is that some of them can support redox cycling of specific compounds. So the mechanism is that some chemical structures can be reduced by a strong reducing agent such as DTT or TSEP, and then be regenerated by reacting with oxygen. Because the reducing agent is in excess, this cycle can go round and lead to the generation of significant amounts of superoxide. The superoxide then gets converted to hydrogen peroxide, and the hydrogen peroxide can non-specifically deactivate the target enzyme. At this point, a number of reactions can occur. An oxidized cysteine can react with another thiol from another cysteine to form a disulfide bridge, which can potentially be reduced by the reducing agent. Alternatively, a number of further rounds of oxidation can occur. And because this really complicates the assay system, it's really important to be able to detect these compounds early on. An assay is well described in the literature to measure the production of hydrogen peroxide caused by the reaction of redox cycling compounds with reducing agents. This assay relies on the oxidation of phenol red by hydrogen peroxide and gives an absorbance readout. This article in the literature describes how the strong reducing agents DTT and TSEP lead to the generation of significant concentrations of hydrogen peroxide, whereas the weak reducing agents glutathione and beta-mercaptoethanol do not. For this reason, glutathione is preferred when working with targets with redox liabilities, as beta-mercaptoethanol is unpleasant to work with. In addition, different enzymes have different sensitivities to reducing agents. So some enzymes, such as target A on the left-hand side, are quite robust and will be active in the presence of various reducing agents, whereas other targets, such as target C on the right-hand side, are much fussier. And so we find it's very useful to test all the different reducing agents up front during assay development in order to be aware of what the possibilities are later down the line. We also bear in mind the possibility of redox and thiol competition when we're interpreting our data. Moving on to the mechanism of covalent inhibitors, and we'll start with a refresher on the mechanism of reversible compounds. This is a simple reversible one-step reaction where the enzyme and the inhibitor form a reversible complex with a forward rate K1 and a reverse rate K-1, and the equilibrium binding affinity Ki with a lowercase I is just the ratio of these two rates. In terms of progress curves, we optimize our assays so that the production of a product is linear over time, and as we increase the inhibitor concentration, the rate decreases, but the curves maintain their linearity. The consequence of this is if we measure the IC50 at different time points, assuming that we have enough signal to noise to achieve a robust signal, these IC50 curves overlay. The IC50 is constant over time. For covalent compounds, the first step of the reaction is identical, but then there's a second step, an irreversible step, where the rate is the K-inact, or the rate of inactivation of the enzyme, which leads to the covalent complex. The parameters now used to describe the kinetics of a covalent compound are twofold. The first is the K-inact, but the second is a Ki with a capital I, and this describes the rate of formation and breakdown of the non-covalent intermediate, and so it's a function of K1, K-1, but also K-inact. It's more analogous to the Km that describes a substrate, and it's worth bearing in mind that this Ki with a capital I is therefore different from the equilibrium binding affinity with the lowercase I. In terms of progress curves, as we increase the inhibitor concentration, the rate is decreased, but not in a constant fashion. It's a time-dependent effect, and so we get this characteristic curvature of the progress curve. The knock-on effect is that if we measure the IC50 at different time points, there is now going to be a decrease in the IC50 over time. The compound will appear more potent as time goes on. This means that it's really important to measure or to characterize covalent compounds in a kinetic way, and this is why we like the PHERAstar FS. We find it particularly user-friendly for kinetic assays. The injectors allow us to study fast-binding kinetics, which we find useful, especially in early assay development. The Mars software allows us to do bespoke kinetic analysis, and we use that hand-in-hand with PRISM. And later on, we look to automate our analyses, and typically we do that using activity-based. Early on, we triage our compounds in high-throughput assays, and we use the stackers on the PHERAstar to do that. We're currently in the process of upgrading our PHERAstar FS for a PHERAstar FSX, and we're looking forward to being able to use the additional capabilities that Catherine described earlier. This is an example of a typical triaging cascade for covalent compounds. We have a subset in our library of approximately 700 covalent compounds, which we can screen in the IC50 Shift pre-incubation assay. I'll go on to describe that in more detail, but that's a crude assay that we typically put at the beginning of the cascade. We can follow this up with more detailed mechanism of inhibition assays, such as the KNAT-KI assay, or the jump dilution reversibility assay. And then we go on to use biophysical methods to get even more information on these compounds using intact mass analysis, peptide fingerprinting, and X-ray crystallography. So first of all, the IC50 Shift assay. The aim of this assay is to identify time-dependent inhibitors. As I mentioned earlier, it's quite a crude assay, but it's also high throughput, and it doesn't distinguish between irreversible and slow-binding compounds, and so follow-up is definitely required. The principle of this assay is to determine the IC50 after different pre-incubation times of enzyme and excess compound. A shift to the left of the IC50 curves over time indicates time-dependent inhibition. We perform the analysis in activity-base and calculate the 99% confidence intervals, and if these intervals are non-overlapping, then we consider the time dependence to be significant. It's important to ensure that the relevant species are present during the pre-incubation. For example, if we're looking at uncompetitive inhibitors which prefer to bind to the enzyme substrate complex, then the substrate has to be included in that pre-incubation, and perhaps there's a second substrate that can be used to initiate the reaction. The jump dilution assay is a more mechanistic assay that looks at compound reversibility. The principle of the assay is to pre-incubate the enzyme at 100 times its final concentration, with the compound at a concentration of 10 times the IC50. This is followed by a dilution of 1 in 100 into substrate to give a final enzyme concentration of 1 times, and the compound concentration is now 0.1 times the IC50. Because the dilution was performed into substrate, this initiates the reaction, and we can follow the product formation over time using a kinetic assay. The assay has been optimized to ensure that the rate of the enzyme reaction is linear over the time course of the assay. For a fully reversible compound, we expect 9% inhibition, as the compound concentration is now 0.1 times the IC50. For an irreversible compound, we expect 91% inhibition, as the inactivation occurred during the pre-incubation. For a slowly reversible compound, we expect something in between with this characteristic curvilinear plot due to the time-dependent process. It's worth bearing in mind that this assay can use quite a lot of enzyme due to the pre-incubation being performed at 100 times the final enzyme concentration. And so typically, we run this assay downstream of the IC50 Shift assay. Follow-up is required to distinguish between covalent compounds and slowly dissociating inhibitors, as they can look very similar. There are a number of considerations to take into account with this assay. The bottom of the assay can be higher than usual due to the pre-incubation with 100 times the enzyme and 10 times the IC50. At this point in the assay, we still want the compound to be in excess of the enzyme, otherwise this could lead to misleading results. The pre-incubation time, 30 minutes is standard. However, longer pre-incubation times may be required if the K-inact of the compounds is slow. Compound solubility at 10 times the IC50 might be an issue. If that is the case, it's sometimes possible to optimize the assay to increase the DMSO content during the pre-incubation, and this can then be adjusted during the dilution. Evaporation can also be a concern for very long assays with long pre-incubations and long kinetic phases. And we sometimes use mineral oil to reduce the evaporation. However, partition of lipophilic compounds can be a concern. The K-inact Ki assay is another mechanistic assay that can be used to determine the kinetic characteristics of covalent compounds. So this is a reminder of the mechanism that we looked at earlier. And the curvature of the plot from these, we can extract a K-obs, and we then plot this K-obs against the inhibitor concentration, and from this plot we can extract the K-inact and the Ki. A kinetic assay is required to obtain a reliable K-obs measurement. And the ratio of K-inact over Ki is a measure of both affinity and reactivity, and a larger ratio is indicative of efficient inactivation. In practice, as the inhibitor concentration increases, it becomes more difficult to measure the K-obs. So an example is here, a real-life example. In the left-hand panel, we can see a snapshot IC50 at 3 hours. This particular compound has a solubility issue at the top concentration, so that point's been excluded. The next highest point generates nearly 100% inhibition, and in the time course we can see that that's nearly overlaid with the no enzyme control, and so the signal to noise for that curve is very small. The knock-on effect is that the K-obs measurement is inaccurate, and it's not unusual to see the K-obs tail off and decrease at high inhibitor concentrations, and we typically exclude those points as we feel they're misleading. We've automated the analysis of these assays as we found it was a rate-limiting step in the process. We upload the .dat file from the PHERAstar into ActivityBase, and we've automated some of the analysis criteria to speed up the analysis. We still manually inspect the controls to ensure they're as we expect. We also manually expect snapshot IC50 curves to ensure we've got a sigmoidal concentration response. ActivityBase calculates the Z prime, and we make sure they're greater than 0.5. The time courses for the test compounds are fitted automatically, and if the R-square of the fit is greater than 0.9, then the K-obs is reported. The K-obs is plotted against the inhibitor concentration, and if the fit is greater than 0.9, the K-inact and the Ki parameters are extracted. If saturation isn't reached, then we can still estimate the K-inact over Ki ratio from the slope of the curve. In terms of biophysical methods to study covalent compounds, we occasionally get asked to look at covalent compounds by biocall. In this experiment, we were looking at USB7, and this compound I'll discuss in more detail during the case study. The principle of this technique is that the enzyme is covalently immobilized to a chip, and then the microfluidics flow over the compound. We're looking for a change in mass at the surface, which allows us to monitor in real time the association and dissociation of the compound. In this particular example, the association was 60 seconds, and over that time we saw a concentration-dependent increase in mass, which allowed us to monitor the association of the compound. And because it was covalent, we were expecting it not to dissociate, so we were expecting no change in mass during the dissociation. In fact, what we observed was quite a fast dissociation, quite characteristic of a reversible compound. And this can be explained because of the relative kinetics of the reaction. So the K-inact, the rate of inactivation for this compound is quite slow. And so the half-life of inactivation, the time taken to form a covalent bond with half of the enzyme molecules available, is nearly 40 minutes. And because the association phase is only 60 seconds, over that time course we can calculate that we'd expect fewer than 2% of the complexes to form a covalent bond, which explains this profile. If we look at the reaction mechanism, what we're actually measuring over that time course is the first reversible phase. So the biocall is not ideal for triaging covalent compounds. If we're doing quite a crude analysis, and if we did more sophisticated fitting, we could tease out the complexes which have formed the covalent bonds from those that haven't. But if we're just doing quite a crude analysis, then it would preferentially detect compounds with a K-inact. Also, if substantial covalent bond formation has occurred, then a new chip or new surface is required for subsequent compounds. For this reason, we use intact mass spectrometry as the next step in our triaging cascade. In this technique, enzyme and inhibitor are incubated to form a complex, and then electrospray ionization mass spectrometry intact mass analysis determines whether the compound is covalently bound and how many molecules of compounds are bound. In this example, the first peak highlighted in purple shows the expected mass of the unmodified target, and the second peak highlighted in red corresponds to the mass of the target plus the mass of the covalent compound. With a very simple system with just two cysteines, for example, point mutations can be used to determine which cysteine is targeted by the compound. In this example, cysteine 1 and cysteine 2, we don't know which one is being targeted by the compound, and peak A is the unmodified target, and peak B corresponds to the target modified with a covalent compound. In the first mutant, cysteine 1 was mutated to an alanine, and the peak was retained, suggesting that binding was occurring at cysteine 2. When the complementary mutant was done, the peak disappeared, indicating that the compound was indeed targeting cysteine 2. When the system is more complicated and more cysteines are involved, then peptide fingerprinting is a powerful technique to identify the site of covalent modification. Again, the enzyme and the inhibitor are incubated to form a complex, but this time a tryptic digest is performed to generate peptides. We typically prefer trypsin over elastase or chymotrypsin, as we find this gives us higher coverage, and we've discovered that reduction alkylation also improves coverage. In this example, reduction and alkylation improved our coverage from 80% to over 99%, and critically, without reduction and alkylation, only 4 out of the 12 cysteines in the proteins were covered, and this increased to all 12 cysteine following reduction alkylation. This is an example of what the output of this technique might look like. And so in black are the residues, the amino acid residues of the enzyme, and in orange this shows the peptides which are covered, and so these are detected by mass spectrometry. In green, we can see the modified peptide, and in this example, the peptide that was modified only had one cysteine in it, so we were able to pinpoint the site of modification. It's worth bearing in mind that the ratio of compound to enzyme might affect how many cysteines are modified, and in this example, when we increased the ratio from 2 to 1 to 10 to 1, we saw a second cysteine being modified. And the specificity of covalent compounds is really important, and a literature method that's been well described to determine how reactive the compounds are is the glutathione reactivity assay. Glutathione is a natural reducing agent, and there's approximately 10 millimolar of it present in cells, and so it's a good molecule to assess how non-specifically reactive the compounds are. In this method, the modification of glutathione by covalent reactive groups is monitored by mass spectrometry, and this paper has tested a number of different warheads and ranked their reactivity. We've applied these techniques to the field of deubiquitinases. At Cancer Research UK Therapeutic Discovery Laboratories, we're part of the DUB Alliance, which is a research initiative to discover tools, technologies, and therapeutic drug candidates against DUBs. This is a superfamily which contains 5 families of cysteine proteases and 1 family of metalloproteases. We're particularly interested in the USP family, which stands for ubiquitin-specific proteases, as these contain many putative drug targets. In particular, USP7 is an attractive drug target. It deubiquitinates MDM2, which itself ubiquitinates P53, leading to P53 degradation. And so the rationale is to inhibit USP7 to prevent the deubiquitination of MDM2 and promote P53-mediated repair or apoptosis, which prevents tumor progression. Until now, USP7 has proven challenging to drug. However, the catalytic cysteine is amenable to a covalent approach. A high-throughput screen of 500,000 compounds was performed at Forma Therapeutics, and a series was identified which had reversible and covalent representatives. We developed a KNAC-KI assay to characterize these compounds, and this was based around a ubiquitin rhodamine substrate, where the rhodamine is linked to ubiquitin via an isopeptide bond, which is cleaved by USP7, releasing the fluorophore and leading to an increase in fluorescence, which can be measured kinetically. We validated the assay using a reactive covalent substrate probe, which is formed of ubiquitin linked to a vinyl methyl ester warhead. This is a very reactive species, and so we characterized it using the injectors to make sure that we didn't miss the beginning of the reaction. We obtained the characteristic curved plots from which we were able to extract the KOBs, which we plotted against the inhibitor concentration to determine the KNACT and the KI. The KNACT can be converted to an inactivation half-life using the equation here, the natural logarithm of 2 divided by the KNACT, and this corresponds to the time required for the compound to inactivate half of the enzyme molecules. In this case, it's very rapid, only 19 seconds. We expected our compounds to be much less reactive, so we reconfigured the assay to run linearly over 3 hours. And this graph shows the evolution over time of the KNAC-KI ratio as the chemistry evolved. We initially selected a ratio of 10 per molar per second, as we found that below that the data were less robust, and we refined this cutoff to 30, which we validated by X-ray crystallography, and we found the compounds we selected, which had a ratio greater than 30, also showed a covalent bond in the X-ray structure. Interestingly, a compound that we were expecting to be covalent that showed no increase in the KOBs, when the X-ray structure came back, we found that the warhead had flipped and that the compound was indeed binding reversibly, and we felt this validated our assay really well. So the output of this campaign was this covalent inhibitor, FT827, which shows here the progress curves and the K-opt plot. And the crystal structure here shows the covalent bond to the catalytic cysteine. These inhibitors are highly selective, and this is in part explained by some of the biology of USP7. So this is a crystal structure that shows ubiquitin in gold, the switching loop in red, and the catalytic triad. And unlike other USPs, the binding of ubiquitin is required in USP7 to align the catalytic triad. In the inactive form, the switching loop is in the in position and the triad is misaligned. And when ubiquitin binds, and you can see the tail of ubiquitin coming in here below the switching loop, this forces the switching loop into the out conformation, which aligns the triad. The inhibitors that we describe bind to the inactive apo form of USP7, which imparts selectivity over other dubs. And in this figure, you can see a panel of dubs run in an in vitro assay, and the compounds that we describe are highly selective for USP7. These novel inhibitors also show promising effects in biomarker, phenotypic, and xenograft models. And we published this at the end of last year in Nature. The learning points from today are that covalent compounds offer advantages in terms of drug residency, and carefully tuned reactivity of the electrophilic warheads can minimize toxicity concerns. Assay development should take into account the effects of thiol-containing reducing agents. IC50 Shift, jump dilution, and KNAT-KI biochemical assays enable compounds to be triaged. Covalent modification can be confirmed with biophysical methods, such as mass spectrometry and crystallography. And the Dub Alliance has identified potent and highly selective USP7 inhibitors which target the unique auto-inhibited apo form of USP7. These show promising biomarker, phenotypic, and xenograft responses, and we're continuing to explore the biology and the disease positioning potential of these novel inhibitors. This leaves me to thank all the members of the Dub Alliance, as well as my colleagues at Cancer Research UK TDL, Catherine Wark at BMG, and Select Science for hosting the webinar.
Thank you, Agnes. Let's move on to the last part of today's webinar, the question and answer session. So, the first question I have here is, do you remove redox cycling compounds from the HTS screening library?
We typically don't remove compounds once we've identified them as redox cycling, because we found it's more work to cherry pick compounds out of the library than it is to flag them. So we flag them in our database, and then if they appear again for a different screening campaign, we can remove them immediately without wasting any time on them.
Lovely, thank you. The next question is, which reducing agent do you routinely use for IC50 Shift assays?
There's no hard and fast rule over which reducing agent we use. We tailor it to each target. We do try and avoid the reducing agents that contain thiol groups, and so if possible we'll use TSEP when we're working with covalent inhibitors, but not all enzymes tolerate that, so it's on a case-by-case basis, and it's important just to bear in mind the other effects that might be occurring in the well at the same time.
Thank you very much. The next question is, have you observed more than one molecule of compound binding covalently to the enzyme?
We have observed that using the intact mass spectrometry. We've had an example where we've had two copies of the molecule binding, and further studies were required to make sure that, to determine whether that was a specific effect of two binding sites, or whether one of them was non-specific. It's important to try and work out what's going on at that point, and ideally X-ray crystallography answers that question.
Thank you very much. The next question is, what is the dead volume of the injectors on the PHERAstar FSX system?
I guess I'll take that question, Agnes. So in terms of the PHERAstar is very reagent saving. You really only need a maximum of 1 ml, including your injection volume. And with us able to inject down to volumes as little as 1 microliter and above, it does mean that, there's very little less than sort of 30 microliters ever left inside the instrument once it's been sort of, washed out or used. So it can be very reagent saving if you're wanting to do your sort of enzyme kinetics utilising the injectors. You don't need to use an awful lot of material.
Fantastic. Thank you very much, Catherine. What differences would I see when measuring fluorescence on the FSX compared to the FS? Okay, so with the PHERAstar FSX, the sort of level of an improvement that you might see for fluorescence could be up to 4-fold. Our limits of detection for fluorescein are down to 0.15 picomoles for, say, fluorescein, for example. So that means in terms of miniaturization, for standard assays, that might improve. Certainly throughput. Read times as well have improved for these types of assays. So when measuring enzyme kinetics, you might achieve faster read times, basically allow you to get more readings in. Thank you very much. We just have time for one more question, which is, what kinetic analysis calculations can be done in the PHERAstar software?
Well, we might both be able to answer this. The PHERAstar can use sort of Michaelis-Menten, Lineweaver-Burke, a whole variety of different analysis plots, basically, if you like. KM, Vmax can be obtained from the system. So I think that's things your team are investigating at the moment, isn't it? More usability of the software.
Yeah, we find Mars is particularly good with the kinetic readouts, and it saves a lot of time in manipulating vast amounts of data. If we try and do it with Excel and Prism, we can spend a lot of time, and we find if we set up templates in Mars, it can save us a lot of time long term.
Yeah.
Fantastic. Well, that's all we have time for today. Thank you very much to Agnes and Catherine for today's informative discussion and presentation, and thank you to everyone for joining us online and for your interesting questions. I hope that you found this to be a worthwhile session. If we didn't manage to answer your questions, we will follow up with you personally, and if you have any more questions, please feel free to email them to me at editor@selectscience.net. If you would like to listen again to today's webinar or invite a friend to listen, it will be available on our website shortly. Goodbye, and thank you once again.
Thanks. Bye-bye.